Howdy, welcome to Chapter 3, Prokaryotes, Part 2. So this is the prokaryotic cell wall. This is really important in understanding all the lab work, whether it's virtual or you're in the classroom, but understanding all the lab work that you will do in staining microbes. So we went over this cartoon in the last video. So this is a transmission electron micrograph. This is actually, this half is the real or the actual transmission electron micrograph, so it's in gray. And then this other half, they've sort of outlined it in a cartoon form and colorized it. So we're going to be zeroing in on this structure called the cell wall. And the cell wall is between this outer sticky layer called the glycocalyx and the inner, this blue line right here, the inner cell membrane. So the cell wall is in between the glycocalyx. The cell wall is in between the glycocalyx and the cell membrane. So here's our bacterium. This would be a bacillus. Cut a rectangle out of that, and then you can see that this sort of brick color, or whatever color that is, tan, sand, I don't know. That's the peptidoglycan layer. That's peptidoglycan makes up the cell wall. And then deep to that peptidoglycan layer, or the cell wall, is the cell membrane, the phospholipid bilayer. And then see this little square here they cut out of the intermediary cartoon, and then they blow it up so we can see what the bacterial cell wall looks like. This is important to note, because we're going to talk a lot about gram-positive and gram-negative cells, but this is a gram-positive cell wall. Gram-negative cell wall looks different, and you might remember that from the last video if you watched it. So superficially, you have the cell wall made up of peptidoglycan, which are these little brick structures, and they have a more intricate molecular structure we'll talk about in a minute. But within this peptidoglycan, you have these acids. So lipoteichoic acid is this guy, and then the smaller ones are just teichoic acid. And then deep to the cell wall, you have your phospholipid bilayer. So here's the hydrophilic heads on the inside and outside, and then the hydrophobic tails, or yellow tails, in the middle. And then here's an integral protein going through the phospholipid bilayer of the cell membrane. This is the molecular structure of teichoic acid. So P, you remember, is phosphate, oxygen, carbon, hydrogen. So you don't have to know that for the test, but that's what it looks like chemically. This cartoon that I just popped on is a gram-negative bacteria. So you can see, this isn't, yeah. Oh, I see what they did. So this is the peptidoglycan layer of the cell wall, and you can see it's thinner than the peptidoglycan layer of the cell wall in a gram-positive cell. And then you have this, here's the inner phospholipid bilayer, the cell membrane, just like in a gram-positive cell, with some integral membrane proteins, but superficial to the cell wall. So if you look at gram-positive, look superficial to the cell wall up here, there's really nothing above it. You just have some teichoic acid, lipoteichoic acid sticking out, but there's nothing up here above the cell wall. But in a gram-negative bacteria, look what you have. You have this outer membrane of the cell wall. So you've got the lipid structure, and then you have these molecules called LPS, or lipopolysaccharides. So all these kind of green things. I don't know what they look like. I don't know. Some kind of old bent cactus or something, coming out of the peptidoglycan layer, or actually it's superficial. The outer membrane is superficial to the peptidoglycan layer of the cell wall. And then here's the cartoon that shows what that peptidoglycan is made of. It's a sugar backbone, and it's got these subunits, NAG and NAM. So I don't know if you guys remember this, but in Vietnam they used to call it Nam. So this Nam, and then Nag, Nam and Nag. That's the molecular structure of the peptidoglycan, which is part of the cell wall. So peptidoglycan covers the entire surface of the bacterial cell. And we said this earlier, there's two basic types of bacteria, gram-positive and gram-negative. And then here's some details. Peptidoglycan. So peptido is protein and glycan is sugar. So they're alternating sugar and peptides. So these little balls here, these are the amino acids of the peptide. And then this shape, this is a sugar. Gram-positive cell walls contain, like I said earlier, contain this really thick layer of peptidoglycan. This is going to be really important when you stain for gram-positive. That's the first stain you do in any lab test in micro. You stain to see if your bacteria, let's say you don't know what the bacteria is, you stain to see if it's gram-negative or gram-positive. So you have to know, when we talk about staining, keep in mind gram-positive cells have this really thick peptidoglycan layer in their cell wall. Gram-negative cell walls, however, have this thin layer. So here's the thin layer of peptidoglycan in their cell walls, and they have this outer membrane. And the outer membrane has an inner leaflet and an outer leaflet. So this would be the inner leaflet, and this part would be the outer leaflet of the outer membrane. And also in the gram-negative cell, there's no teichoic or lipoteichoic acid in the gram-negative cells. Gram-negative cells will stain pink in the gram stain. So when you do a gram stain, these guys will show up pink. And then I told you this molecule out here is LPS, or lipopolysaccharide. So lipo is lipid, and polysaccharide is many sugars. So LPS consists of lipids and sugar. And then the lipid portion of lipopolysaccharide is a specific lipid called lipid A. So this is in gram-negative bacteria. So when these gram-negative bacteria die, this LPS, which is a type of endotoxin, will be released by the dead bacteria, and then your body is flooded with LPS, which is toxic to you and can kill you. The outer membrane can help prevent movement of some drugs into the bacteria, so they're harder to kill because they have this outer membrane. Gram-negative bacteria are harder to kill. Okay, so here's a gram stain. We talked about this gram stain for bacteria. So if we come over here with the key, so this kind of bluish purple stuff, or violet, is crystal violet. The red in the key is iodine. No color is alcohol. And this pink color is safranin. So in a gram stain, so here's your bacteria. So look, check it out. You have some rods, you have some cocci in there, or rods is the same thing as a bacillus or a bacilli. And the first thing you do in a gram stain to see if you have what kind of bacteria you have, you apply this crystal violet, which is a purple dye. And then you will rinse, you'll do some rinsing, and then you'll apply iodine, which is a mordant that locks the color in. And then you'll do an alcohol wash. You'll decolorize all the bacteria. Well, you'll decolorize the gram positive bacteria. Well, I'm sorry, you'll decolorize the gram negative bacteria. And then you'll stain with this safranin, which will stain the gram-negative bacteria pink, while the gram-positive bacteria, because they have that thick peptidoglycan, will retain the crystal violet dye even after trying to wash it out. So the correct order of steps in a gram stain: you do the crystal violet, then you do grams iodine, the mordant locks color in, then you decolorize, then you do a safranin counterstain. So the two stains are crystal violet and safranin. So it just shows you in this cartoon, when you put on the crystal violet, everything stains purple. You can see that here. So if you are a rod, or aka a bacillus, or if you are a coccus, you'll stain purple with the crystal violet. Then you put on iodine, which is a mordant that will sort of lock in the color. And then you use this alcohol step to decolorize. So because these rods are gram-negative, they don't have that thick peptidoglycan layer. In the destaining or decolorizing step, I should say decolorizing step, they lose that purple color. But because these gram-positive cells that are purple, they have such a thick peptidoglycan layer, they hold on to the crystal violet. Then you give a counterstain, safranin, and while the gram-positive bacteria will take up some of that stain, the gram-negative will take up that stain, because remember, they were decolorized, and they'll be pink. So the gram stain is based on, and it works because of the differences in the cell walls of gram-positive, gram-negative bacteria. And we just said this, gram-positive cells have a thicker peptidoglycan than gram-negative. And I told you this too, in gram-positive cells, the crystal violet dye is retained in that thick cell wall. So gram-positive's got this, here's a cartoon of the thick cell wall that retains the crystal violet stain even when you try to decolorize it. So gram-positive cell walls. So special case, Mycobacterium, that's tuberculosis, TB, and leprosy. They're gram-positive, but they have these waxy lipids in their cell wall, and they're difficult to stain. So they use a special type of stain. It's called an acid-fast stain. So you can't stain these. They are gram-positive, but it doesn't show up very well. So you use a different stain called the acid-fast stain. And in the acid-fast stain, it shows the steps down here. Apply the primary stain, carbonyl fusion. You give that for 30 seconds. Then you heat fix the cells. So this is supposed to be a flame here. So you're fixing the cells, all the parts of the cell in place. And then you decolorize. And then you apply this counter stain called methylene blue. And if you are an acid-fast organism, you didn't get decolorized. But if you are a non-acid-fast organism, you were decolorized, and then you were counterstained with this methylene blue. Okay, so tuberculosis, they call it an acid-fast bacterium. So you have to use an acid-fast stain if you want to see them. So the chemical and we said this earlier, but the chemical in some bacterial cell walls, like the bacteria that cause tuberculosis, they're resistant. That mycolic acid makes them resistant to decolorization after acid-fast staining.